Hey, ladies and gentlemen, welcome. We are going to circle back to chapter 25 and talk about the tail end of this chapter. I like to split it out because it felt more like the end of chapter 25 was circling into some more evidence for evolution as opposed to really truly relying on the history of Earth and just how life got here in the first place. Um, so let's talk a little bit about this idea of evolution of development. So we've got life. Life is on land. We've got different metabolic processes like the oxygen revolution taking place. But then how do we get all of these changes in these body forms that we end up seeing as species evolve from just being these single-celled prokaryotes to these multicellular complex organisms? So they typically end up changing based on sequences and the regulation of certain sets of developmental genes, especially when we start talking about these genes in organisms like mammals. So being able to study these genetic mechanisms of change really provides an interesting insight into this large-scale evolution that changes that occurred as we study through the fossil record and even up into modern organisms. So what we're really looking at is an entire field called EvoDevo, or evolutionary developmental biology, where we're looking at these genes that are specifically programmed to control the rate of development, the timing of that development, the spatial patterns that we see in these complex organisms as they go from fertilized egg to embryo to fetus to infant to adult. Um, and so we're looking at this idea of heterochrony, these evolutionary change in the rate or the timing of these developmental events that really seem to have a huge impact on the way that the body forms, the morphology of that adult. So if we look at just an example, the skulls of humans versus chimpanzees. And it really comes down to an interesting gene change in the rate of when our jaw muscles really sort of take form that then also alters when those fontanelles, those soft spots, fuse in our cranial structure. So the jaw muscles in a chimp are much stronger, much thicker, and as a result, force the closure of their soft spots, those fontanelles, in young chimps much earlier on in development, where us in humans, it's not quite so strong, and as a result, allowed for those soft spots to stay open longer. They don't fuse as early. And so we can see this pretty significant change in cranial size as a result of that change in that particular gene structure, allowing for theoretically us to have a larger brain size and the ability for some of the higher order cognitive function that we have above a chimp. Heterochrony can also alter the timing of reproductive development relative to other sort of non-reproductive organs. And so you get this interesting idea in these salamanders, for example, that actually retain this tadpole characteristic, including their gills, even though they are at this point sexually mature adults. They still sort of look like the juveniles of their ancestral species. And this is this idea of platomorphosis. So this rate of reproductive development is actually accelerated as opposed to comparing that typical rate to those somatic cell development, the rest of their body development. And so although they're sexually mature, they retain these features of juveniles that we would have typically seen in their ancestral species. So looking like a juvenile, but being completely capable of sexual reproduction because we've changed the rate of development of certain characteristics in their body plan over evolutionary history. One of the things that happens a lot when we look at changing, mutating the rate of developmental mental genes is these changes in spatial patterns, the way that the body is organized, the placement of certain parts, and most of this often occurs in the set of genes that are pretty commonly that they exist quite widely across a lot of different organisms, and that's this idea of these homeotic genes or Hox genes. And these Hox genes are a set of genes that have gotten duplicated from one species to another, thus taken on new roles. Um, but the idea is that they determine basic features and where they're supposed to be arranged. Everything from the wing and leg development in a bird or the arrangement of flower parts to even us, we also have these Hox genes as well. So they are a class of homeotic genes that provides positional information. So it tells the body where the head should be, where the tail should be, where your limbs should be. And the way that those Hox genes work, if they're expressed in the wrong locations, to the wrong concentrations of molecules that they produce, whatever the case might be, we end up with different parts in different places. And we've done a lot of mutational studies on these genes in certain organisms. We'll come back to them when we talk about gene expression again, specifically studying them in fruit flies and watching them have two heads or legs where their antennae should be. So we've done quite a bit of research on how these Hox genes work by mutating them and finding out what happens as a result. And so we believe that changes in those spatial patterns, right, may be associated with alterations in these genes. So this particular example is looking at vertebral structures in this particular set of organisms, this particular set of fish, to get us from sort of this really truly ancestral organism to jawless fish to ultimately having jaws. And it's a matter of duplication of these Hox genes and then some mutations within those duplicated copies that allowed potentially for the evolution of this jaw structure in these fish. We think this may be a contribution to that huge diversity that showed up during the Cambrian explosion. Developmental genes may in fact play an important role in how we got there. So looking at changes in those developmental genes and the morphological forms that are associated with it. So we look at something like the Drosophila, which are your fruit flies, and this little guy here, sort of a shrimp-esque organism, and we look at a gene mutation that occurs here in Hox gene 7 about 400 million years ago, and it has altered the patterns that we see in their limbs. So new forms most likely come from gene duplication events that then produce some additional mutations within those duplicated copies that then produces some additional developmental changes in their body plans. So it's a possible mechanism for how these are in fact working. So this particular Hox gene 7 contributes to the number of legs in this crustacean species. We've actually been able to demonstrate this in a lab by changing that UBX gene and turning it off partway through leg development. We get that reduction in the number of legs from that crustacean species to the fruit flies. So we're able to actually demonstrate this in a lab. We think a lot of those morphological changes are a result of regulation of developmental genes rather than the actual sequence itself. So we may not necessarily be seeing a change in the actual A's, T's, C's, and G's in the gene, but by regulating when that gene gets turned on and off. So we'll talk about genetic switches that are actually upstream of the gene itself that can then alter when that gene gets turned on, when it gets turned off, how long is it on, how long does it stay off, when does it get turned on and off. All of those things will have a huge role in the early development of organisms, where things are located, how many limbs do they have, things like that. And so take a little time to go through this particular experiment in your textbook about the three-spined sticklebacks. When we see them in certain environments, lakes in particular, they've actually got fewer spines than their marine relatives. And so that's led us to taking a look at the rate at which the particular developmental gene that's associated with those spines, when does it get turned on and off and for how long? And so the gene sequences, again, remain the same, but the regulation of that gene expression is different in these two groups of fish, as a result leading to some morphological differences.